So next speaker will be Marion Schmicke, and she will give us some insight into modeling of animal cells.
Yeah, thank you very much for this kind introduction. I just need my presentation. So I designed the presentation to give you a rough overview about how to model animal cells for research, and particularly hepatocytes, as my work group worked with hepatocytes, and we did in the BOFREC project. Yeah, why hepatocytes? You may all know, if we speak about productivity and livestock and cattle in the BOFREC project, the liver is the main central metabolic organ, and therefore it's very important. The liver also facilitates endocrine functions, for instance, linking metabolism and fertility by producing fibroblast-like growth factor 21, for instance. And it also serves as an immune organ because only 80% of the liver consists of parenchymal cells, hepatocytes. 6.5% of the liver consists of immune cells, so lymphocytes or resident macrophages like Kupffer cells, and 13.5% of the whole liver is about sinusoids, so and small capillaries. So this is space where the blood is drawn through, and the biocanonicules. So if we have a closer look at these cells, hepatocytes are epithelial cells, but they are very specific. Normally, epithelial cells do have one basal layer and an apical side and lateral cell-to-cell contacts. Whereas in hepatocytes, we have two layers of a basal membrane. They are laying in sheets in a single layer in the liver, and the apical sites form the bile canaliculi, so a very special forming here. The non-parenchymal cells are physiologically not in a direct contact to the hepatocytes, but in between there is a distal space and the capillaries where the blood comes through. So this is important if we would like to model the cells, of course. And if we have again a closer look, you see on the right-hand side here that we have differences with regard to the position of the cell to the central vein or the periportal side. And this makes a difference in the hepatocytes. So periportal hepatocytes do have a higher content of enzymes which are involved in glycogen degradation and also in gluconeogenesis. Whereas the paravenular hepatocytes have a higher content of enzymes involved in glucolysis and glucogen synthesis. So we can say hepatocyte is not equal to hepatocyte, even the physiological cells. Yeah? So we have to have this in mind when we would like to model hepatocytes. So how can we model them? We can do it in a monoculture, so where we only have the hepatocytes in culture. And we were able, within the BOFREC project, to work further and to improve protocols for having hepatocytes in this monoculture. Another option is to put them in a co-culture with, for instance, these immune cells. So as you can see here, the small red dots are Kupffer cells, healthy Kupffer cells, which has phagocyted fluorescence labeled beads, so we can see them. And what we did is I would like to show you that it makes a real difference where you put the Kupffer cells on. So you can, in culture, put them in a direct contact to the hepatocytes. So you have a collagen layer as mimicking a basal membrane, then put the hepatocytes on top, and then on top the macrophages. Or you may have it in an indirect contact, so having a collagen layer, the hepatocytes, a collagen layer, and then on top the Kupffer cells. And as you remember, this is more the physiological way where the non-parenchymal cells are, and this makes a difference. As you can see here, the albumin expression, so albumin is one of the most important protein produced by the liver, it's very specific, and you can see here that the expression of albumin is higher in culture if you have Kupffer cells in the indirect contact compared to having no Kupffer cells or having the Kupffer cells in a direct contact. So this makes a difference. Then you can culture hepatocytes in a monolayer, so just having one collagen layer. Collagen is the main protein of the extracellular matrix, so therefore mimicking the basal membrane. But as I already introduced, it's more physiological to have the cells in between two layers of the basal membrane. So therefore, the 2D sandwich culture is the most physiological way, I think, for hepatocytes. And we were able to establish respective protocols within the BOFREC project in order to do so. And there are other culture options, of course. So for instance, a 3D culture, also hepatocytes are able to aggregate to these 3D spheroids. And we, of course, have the opportunity to have organ-on-a-chip techniques today, where we have this microfluidic chambers, where we can also mimic the blood flow through the cells via these little chambers here. So if we would like to model hepatocytes, we need the hepatocytes, of course. So I would like to give some insights on the source of cells. So within the BOFREC project, we work with primary cells. These cells are ex vivo culture of freshly obtained cells from a multicellular organism. With regard to the hepatocytes, the proliferation in vitro is limited because they are in a growth arrest. But from one liver, we got hundreds of millions of cells, and before BOFREC we had no protocol to cryopreserve those cells because bovine hepatocytes are very sensitive cells. We cannot use the established protocols. So therefore, Sandra Andres, she did it, so many thanks to her. Here she worked on a protocol. We do a two-step collagenase digestion with this liver and then gathered the hepatocytes and established a protocol for cryopreservation. So what we could show is that we definitely need controlled rate freezing for hepatocytes. We cannot freeze them manually, so the viability is much higher if we use controlled rate freezing to prevent the formation of intracellular ice crystals. And also a very common used cryoprotective agent, DMSO, was very toxic to the bovine hepatocytes, interestingly, more toxic than compared to porcine or rat cells or so on. We don't know why the bovine bovine hepatocytes are much more sensitive. And we could show that the addition of trehalose was, as a non-penetrating cryoprotective agent, was better than the others. However, the recovery rate of hepatocytes is low after the freezing process because due to the freezing process, there is downregulation of beta-integrin, and then we have lesser cell-cell contact or contact to the matrix in these cells when we have them once freezed. Then, of course, you heard already, we have the option to immortalize cells so that they are manipulated and maybe cultured for longer periods of time. We know in hepatocytes specifically that the enzymatic liver digestion we use triggers the G0 cycle of the transition of the hepatocytes, so therefore they arrest in this growth arrest. We can insert immortalization genes like HTERT in order to overexpress it, and then we hope to control or leading to a controlled cell proliferation. And we were able to establish in the BOFREC project a specific protocol in order to get, not using a virus, but lipotransfection genes into the cells. We also were able to get HTERT in the liver cell, but this does not lead to an unlimited cell division in the hepatocytes, as they are in the growth arrest. They do not proliferate normally, so therefore additional growth factors or the transfection of oncogenes are necessary to get really a cell line from the bovine cells here. And in the last 2 minutes, I would like to show you two other options where we can get the cells from. The one option is, for instance, we have the HepG2 cells available. These are human liver cells. And these cells were once isolated in 1975 from a male 15-year-old man with liver cancer. So we still, since then, have these cells and they still proliferate. So the question, of course, is if these cells are still the cells from 1975. But we do not have these tumor cells from a bovine species. And another option we are currently working on is using embryonic or induced pluripotent stem cells. So what we can do there is we can culture the embryonic stem cells on feeder cells. They grow, and then you put different factors afterwards to these cells to mimic embryogenesis and induce the embryogenesis and later on get hepatic or bovine hepatocyte-like cells. So there are different stimulation factors here to get at first the definitive endoderm, the hepatic endoderm, and all the steps we have in embryogenesis as an option to get hepatocyte-like cells. Those cells are also able to produce albumin. And this is maybe some closing hint here. The cells, so if you make cells, it's of course important, as Shama says already, that these cells produce the proteins they should produce, or they produce also in vivo. And for hepatocytes, albumin is just a marker, for instance. So therefore, these cells are able to produce albumin, so therefore, they are not hepatocytes, but hepatocyte-like cells. And with that, I would like to thank you for your attention. I'm happy to answer questions, and all the BOFREC partners and Christa for leading this project.
So, thank you, Marion. Question? Hiroki?
Thank you. And I was just wondering, and I had a question, like you kind of mentioned in your presentation that porcine hepatocyte is much easier than bovine cell line establishment and everything. So do you know anything about porcine hepatocyte cell line that has been established, or like by yourself you have an experience with that?
Yeah, thank you for this question. No, I'm not aware about a porcine cell line until now. We have compared the cells from porcine and bovine cells, and even we also work with red cells and so on, and everything what we did, the bovine hepatocytes are very sensitive for cryoprotection, for all these steps, handling them in the lab without really having a clue which specific factors are, what is the background, why the hepatocytes are so sensitive from specifically adult bovine cells. So it's not comparable to fetal cells. So I also have to mention that because some cell lines are available from fetal cells, but fetal hepatocytes are not the hepatocytes from adult cattle, because of course we have the rumen. Later on we have a completely different animal as an adult ruminant. So therefore it's, I think, very important to get cell lines from adult cells and not from fetal cells with regard to the question of your experiment.
Further questions?
There's a question for Doreen. Dominique. Do you want to read it?
Yeah, sure. I want to read it. So Dominique asked if we see any structural aberrations encompassing genes regulating cell proliferations in the cell lines. So I didn't actually look yet at the gene contents of the structural areas, but of course that is something we should look into, because actually it is shown that aneuploidy is actually detrimental usually, but in those cell lines it seems not to be the case. Actually, it's a very complex topic. The aneuploidy in those cell lines actually accelerates proliferation. So cell lines are usually known for a better proliferation efficiency. So that's why we should look into it. Another thing that is influencing that is also the immortalization, because we don't really know, especially for the spontaneously immortalized cell line, what is the driver of this immortalization. Thanks.
Christa?
Yeah, thank you. I was just curious with this HepG2 cell line. It's almost 50 years old now. Has anybody ever tried to sequence this cell line or made this public?
No, I don't think so, and it would be, of course, interesting. We did not did it because it's a human cell line, of course, not a bovine. It would be very interesting, and you also mentioned that there are subclusters already. So, but of course, it's always interesting that we have the cell line nearly comparable for these 50 years, because you see how difficult it is to make a cell line with immortalization processes and so on. And this is a cancer cell, and this proliferates since years. So maybe we need to know the mechanisms behind this spontaneous immortalization processes. Yeah, so we have the stable cell lines from these processes and not from immortalization of primary cells.
Do you know if specific areas of the genome would be more prone for these aneuploidy or triploidy? So that would be maybe from human research, when you're a specialist in cells and cell lines?
No, it's a good question, but I'm not aware about this data. Yeah, it would be very interesting.
In my previous laboratory, we sequenced several different liver cancer types, including HU7 and HepG2 and everything. But I'm a little bit scared, like you kind of mentioned that after the trypsinization and passing like several times, like if the mutations and like specific type of the deletion can be caused. But I think, I don't know, like maybe if somebody knows about this fact, I want to know about this because we are using it.
Okay, thank you. Thank you, Marion.